Hi everyone! Thanks for watching our online presentation of the results from our validation study. This study was conducted by Torrie, Sarah, and myself, Kaylee. Our validation focused on comparing automated extraction using the QIAGEN EZ1 robot to manual extraction and purification of DNA. Specifically, our study evaluated Promega's DNA IQ manual extraction kit versus QIAGEN's EZ1 robot for automated extraction. We wanted to find out which method provided better quantitative results. Operating parameters of the EZ1 robot, like its accurate measurements and its isolation of the samples, are expected to produce an increased yield result for our samples. As outlined in the SWGDAM guidelines, we performed the following studies. We intended to perform testing on both known and mock evidence samples, but due to the supply limitations of the lab, we only were able to complete the known samples, which for our case was saliva on the buccal swabs and blood on filter paper. For the precision and accuracy study, we evaluated the method's repeatability and reproducibility. For repeatability, we evaluated this by comparing the results from each individual team member using the same sample and extraction methods. For reproducibility, we evaluated this by comparing the results of the extraction methods produced for each sample between each of the team members. So for this study, each of the team members were given the same 4 samples, and each sample was extracted both manually and by the robot. So more about the different methodologies that we used for our study. Promega's manual DNA IQ kit requires a more hands-on approach that uses the silica-coated paramagnetic beads to bind to the DNA in our sample solution. These beads are then washed multiple times and the DNA is eluted off using an elution buffer. This method requires multiple washing and transfer steps that could result in decreased yield for available DNA analysis further downstream. On the other hand, the automated EZ1 robot uses an enclosed instrument that is configured to decrease the possibility of contamination or other errors that could result from reagents or pipetting that we could possibly see using the manual method. And unlike the DNA IQ kit, the quantitative data recovered is expected to be increased for using the automated method. For the picture on the left here, this is a picture of Promega's DNA IQ kit. The photo in the middle is of the EZ1 robot, and the photo on the right is of the inside of the robot. So here you can see where the samples and the different tubes go in, and here is where you would place the different reagents. We had a total of 24 different samples that we used. There were 4 samples per team member that were each extracted twice, once using each method. We split these up using sets A, B, and C to represent each team member. So the sample type in the medium that we used is that each team member got 2 samples of blood on filter paper and 2 saliva buccal swabs. Then, like I said before, each sample was extracted twice, once using the manual DNA IQ and once using the EZ1 robot. So for example, the way that we labeled our samples here was that for person A, they had the A1 blood sample extracted twice, the A2 blood sample extracted twice, A1 saliva, and A2 saliva again. For methods and materials, again we used the manual DNA IQ kit from Promega and the EZ1 robot from QIAGEN. Samples were processed for both methods by lysing the cells in a Gill buffer, Pro-K, and DTT solution. The sample buffer Pro-K DTT samples were incubated at 95 degrees C for 30 minutes. After the incubation period, the manual DNA IQ kit samples were then extracted using the different reagents listed below. The time that it takes to process a sample manually using this kit is approximately 70 to 80 plus minutes, including the 30-minute incubation period. After the incubation period for the EZ1 robot samples, the lice samples were placed inside the robot along with the reagent cartridge, which includes the magnetic particles, the lysis buffer, the wash buffer, and the different elution buffer. Once inside the robot, it's smooth sailing from then on, and your final product is a fully eluted DNA purified sample. The time it takes to process these samples on the robot is about 45 to 50 minutes, which again includes the 30-minute incubation period at the beginning. For our further downstream DNA analysis, quantitation was performed on an ABI Prism 7000 instrument using the quantifier human kit that utilizes real-time PCR. Amplification was performed on a 9700 thermal cycler using the PowerPlex 16 hot start amplification kit. Finally, separation was performed using capillary electrophoresis on a 3130 genetic analyzer that used performance optimized polymer 7. For our results, this table shows the quantitation data that was obtained for the 8 different samples that were extracted for each of the team members. As you can see here, for the manual blood samples, DNA concentrations range from 5.77 times 10 to the negative 5 to 1.9 thousandths of nanograms per microliter. For the manual saliva samples, DNA concentrations range from 1.6 thousandths to 17 thousandths nanograms per microliter. For the EZ1 robot blood samples here, DNA concentrations range from 0.179 to 0.494 nanograms per microliter, and for the EZ1 saliva samples, we have 3.52 to 17.6 nanograms per microliter of DNA. Even though our main focus of this study was to look at the DNA yield produced from using the different extraction methods, we did continue on to do qualitative results on our samples. 11 out of our 24 samples produced full or usable partial DNA profiles. No profiles were obtained for the manually extracted blood samples, as you can see in this table here. We did, however, have different variable results for the manual saliva and the EZ1 blood and saliva samples, as seen here. Here we have an example of an electropherogram obtained from a manually extracted saliva sample from team member B. As you can see, the electropherogram shows that this is likely a single source sample, as expected, with 2 or at least 1 allele showing at almost every loci. You may notice down here we have a red X crossing out one of the possible peaks. We use those X's to note places of possible stutter and or pull-up. Again, here's an example profile of an EZ1 robot extracted blood sample from team member B. And here an EZ1 extracted saliva sample from team member C. And these are the types of profiles that we would be looking for to use for forensic casework. After testing, we found that the EZ1 robot extracted significantly more DNA than the manual DNA IQ kit for all samples. For repeatability, the results were relatively inconsistent among the same types of samples for each of the team members. The best repeatability was seen for the EZ1 robot extracted blood. For reproducibility, however, we showed that this was possible in that at least 2 team members had similar results for each sample type. Variable results among the same samples and team members is possible, however, due to the amount of starting material used. While we attempted to use the same amount for each of the samples amongst team members, buccal swabs could have different amounts of saliva and or DNA, whereas errors in measurement and cutting of the blood samples from the filter paper could have caused differences. Our qualitative results were inconclusive due to the lack of comparable profiles amongst sample type, method type, and between team members. Again, here's the table showing the different quantitative results that we obtained for all of our samples. As we can see here, the EZ1 extracted samples produced a much more higher concentration of DNA than the manually extracted samples. Now, as you can see, when comparing samples of the same type among different operators, of the same operators, results were inconsistent, showing that repeatability was not greatly achieved. However, when you look at samples of the similar type amongst the same operators, you can find that there are instances where at least 2 operators had similar results. And again, a lot of these differences can be due to variable or variations in the starting material used for the buccal swabs or the cuttings from the blood filter paper. In conclusion, we found that the EZ1 robot is much better than the DNA IQ kit for DNA yield extraction from both the buccal swabs and the blood cards, or in our case, the blood filter paper. Due to its simpler protocol, it's less processing time, and it's less possibility for contamination, meaning less handling and isolation. We feel that EZ1 robot is better for forensic casework. Even though repeatability was not proven successful in this study, it could possibly be demonstrated if there were a greater sample quantity. In our study, we were only able to use 2 samples of each type for each team member. And third, in our study, reproducibility was shown to be possible, but it could be better demonstrated again with a greater sample quantity. Thank you for listening to our presentation of our sample validation results.